It's my privilege to now introduce Dr. Stéphanie Cherqui, really someone who probably doesn't need an introduction at this point, but I'll just mention that Dr. Cherqui has served on the CRF Board of Trustees for many years. She's now the chair of the Scientific Review Board, which helps decide where research dollars go, and she is a fierce advocate for our community. My boys and I were able to visit her in her lab a few years ago, and I was impressed to see that she has many photographs of people with cystinosis in her office, and I think she's really dedicated to this group. And so she spent, more than 2 decades now doing research on cystinosis. So I'll now turn over the time to you, Dr. Cherqui.
Thank you very much. Thank you for the introduction. As every year, it's very special for me to be, at the Day of Hope. Thank you for all the support, all the kind words. It's very special. So I will give you an update on the stem cell and hematopoietic stem cell gene therapy for cystinosis clinical trial. First, I have to start by my disclosure. So I am a co-founder for Papillon and chair of the Cystinosis Research Foundation Scientific Board. So, as we heard earlier by Dr. Jenkins, the treatment that exists so far are like the cysteamine and the supportive, all the drugs that we have to take to treat the symptoms of cystinosis. The approach we are taking is different. We want to treat the beginning of the issues. We want to be able to reintroduce the default gene that is missing into the patients. And this is what we call gene therapy. So the issue with cystinosis is that this gene is expressed in every single tissues, every single cells. So how do you bring one gene to all the organs? So this is the issue that I was dealing with, because I did my PhD on cystinosis, and I found the gene, and I really wanted to go to the gene therapy. This was my goal. But when I realized, we didn't know at the time that it was expressed in all the tissues. And if I would have known beforehand, I wouldn't have chosen cystinosis as a model. But it was too late. I was already involved with all you guys. I really knew it, and it was like a personal fight. And I was like, okay, we have to find something. So the way we decided to go for the gene therapy is not to introduce the genes randomly, because it would never work. So I decided to take the hematopoietic stem cells, so the bone marrow stem cells that we all have in our bone marrow. And why it's important is that they can live, they can stay in your bone marrow, reside there, and if you have any injury, they can travel to your tissues. So I was like, why don't we use that as a vehicle? And so you can see that the principle of the ex vivo gene therapy is that we take the hematopoietic stem cells from the blood of the patients, and we ex vivo, so in a culture plate, we modify them using, in the case of cystinosis, we use a viral vector, but you can also use gene editing to correct the gene. Then after you do a chemotherapy and you reintroduce the cells, and then, as I said, the new cells become part of the body. They will go into the bone marrow, reside there, multiply there for the rest of the life of the patients. And so these cells, as Stephen said this morning, these cells become blood cells. So of course it makes sense that these cells can treat blood disease. But it didn't make sense that how can we treat tissue degeneration? And so, as again, Stephen, and thank you, Stephen, by the way, for reintroducing. And I'm using your Prime, the Amazon truck, of delivery of lysosomes. So we have a monocyte, and these cells that are part of the blood cells, these monocytes, they have the ability of becoming macrophages into all the tissues, or macroglia-like cells in the brain. So you see that if we do one transplant, the cells that we correct become into the bone marrow, and after, they will target all the tissues for the rest of the life of the patients, and bringing, with this truck, the healthy lysosome to all the tissues. So this was the theory behind it. And so we did all the study in the mice, so I won't show all the study in the mice because we passed that. I'm sure you all want to see the results in the patients. But so what we have shown is that we used a lentivirus vector, which is a safe version of HIV, and we use this vector to introduce the Cystinosis gene into the hematopoietic stem cells. And with that, we did all the preclinical studies. We went to the FDA, and we were clear to be able to start a clinical trial. And this was we started in 2019 after we secured a grant from the California Institute of Regenerative Medicine and from the CRF. And so we have, I am the principal investigator, but we also have Bruce Barshop and Edward Ball. Bruce Barshop is here. I mean, he was here this morning, but for sure he will be there tomorrow. If you have questions, you can ask him. And so the targeted number of patients were 6, because in gene therapy, the number of patients for clinical trial are very small. And this was the first phase of a clinical trial. So the primary endpoints, what we wanted to show is to show that it was safe, because it was the first in human using hematopoietic stem cell gene therapy for cystinosis. So we have to make sure that this was not an unexpected side effect. So that was the goal of a clinical trial, and we had several secondary endpoints to test the cystine content and clinical outcome. So all the patients who started the clinical trial, they had to stop cysteamine 2 weeks prior the stem cell injection, and they had to stop the cysteamine eye drop 1 month after the stem cell injection. If there is no reason to restart cysteamine, they are not restarting cysteamine. So all the data you've seen, unless I said, the patients are off cysteamine. So as I said, all the patients that were coming, they were go through screening, and if they were meeting the criteria, they were starting all the assessment to see all the to estimate the tissue health. And after they go through what we call apheresis, so they have to get the white blood cells when we put back the red blood cells. So this takes like 4 to 5 hours. And when we get enough cells, the cells were transported to UCLA in the room that is called GMP, so they can manufacture cells that can be transported to the patients. And there, they go through the gene modification. If the cells look safe, because we do a lot of testing in the cells, then they are frozen and they come back to UCSD, where we do an infusion. And this is actually our first patient going through the infusion of the stem cells. I mean, I will always remember that day. It was really like giving birth after so many years, because it's actually more than 20 years that I'm working on cystinosis. So to reach that point of what you have done, really going to a patient, was very exciting, but very scary at the time. And it's still scary, but a little bit less now. So I'm happy to report that we have completed the phase 1/2 clinical trial. I mean, we enrolled the 6 patients. So we have 5 males and 1 female, patient 5. The age of patients at the time of a transplant was included between 20 years for patient 1 to 36 years for patient 2. Patient 1 and patient 5 did not have a kidney transplant at the time of the stem cells. All the other patients had kidney transplant. So I'm happy also to report that there is no adverse event or serious adverse event related to the stem cell gene therapy. So this is a very important data. All the adverse events that we had in the trial are mild to moderate and are due to mostly to the chemotherapy, because the patient have to go through chemotherapy. Even if it's a low dose, it's still, there are side effects. It's also due to the underlying disease, cystinosis. And we had one serious adverse event, so meaning that it was required about hospitalization, and this was for patient 6, but it was because of a pre-existing condition. So it's a condition that he had before the stem cell transplant. So to measure the efficacy of a gene modification, what we do is that we measure what we call the vector copy number. So it's the number of vector per cell. And as you can see here, we have a vector copy number, VCN, that are included from 0.6 for patient 4 to 2.9 for patient 6. So this is what we have in the stem cells. But after, we have to verify what the cells become, what is the vector copy number in vivo. And what we do for that, we just do a simple blood draw at the different time point. And you can see that we have 4 of our 6 patients that were with a vector copy number above 1. And I think this is really important because I will show you data after. And actually what we can see now, a trend that when it's above, the vector copy number is above 1, the outcomes are better. Unfortunately, the patient 2 didn't want to continue to be seen at UCSD, so we don't have any data from patient 2 after 1 year, and that's very unfortunate. But fortunately, we have the 5 other patients that continue to come every time, so it's demanding, but they are here, and we have 4 patients out of 6 that are here today, so we'll answer all your questions. So we have the best vector copy number for patient 3 and patient 6. And patient 1 had a drop of vector copy number from 2.8 to 0.6 at 6 months. So this can occur because at 6 months you have progenitor cells that die, and the true stem cells that will stay for the rest of their life really take over. So sometimes you have a decrease like that. But what we have seen for patient 1 is after a progressive increase, and now it seems really stable for many years, like over 4 years, above 1. It is at 1.1 now. We have patient 4 and patient 5, patient 4 and patient 5, who have a vector copy number lower than 1, and is 0.5 and 0.6, and 0.7 for patient 5. And because the outcome, we realize that the outcome are not as good, both patients have been advised to go back on 25% cysteamine. So patient 5 have already started, and patient 4 will start soon. So what about the leukocyte cystine, the white blood cell cystine level? So as you can see, all the patients in the trial, after they got the stem cell transplant, so this was at baseline without cysteamine, this is the white blood cell cystine, you can see that there is a decrease of cystine in the white blood cells, except for patient 4, and that's why we said last time, maybe you should go back on low-dose cysteamine. It's not, there is still a decrease. I mean, I think it's a decrease of about 40%. So the protein is there, and there is action of the protein, but we feel like it's not enough compared to the other patients. We had the same thing, so this is patient 5. She was not on cysteamine here, and you can see also that compared to baseline, they were like still 60% of decrease of a cystine level. So the protein is still there. But you can see with 25% of cysteamine, now she's very low on cystine level. So, and the good news about that is also she was on 25% cysteamine, and the vector copy number doesn't seem to have decreased. So it's not incompatible. That was what we were not sure. As you can see for patient 1, for whom we have the most data, I mean more than 4 years now that is out of cysteamine, we have a feeling at UCSD now, the normal of cysteamine level is for healthy below 1. At UCSD, it seems like, around 2 seems to be good enough. But there are, anyway, there are some difference, in measurement between labs, and you can see that for patient 1, during the last 2 years in his hometown, is actually below 1, for over 2 years, despite the fact that he's without cysteamine. We also do a biopsy, so skin biopsy every 1 year and rectal biopsy every 6 months. So for most of the patients, what we have seen is that there is a decrease of the cystine crystals in the biopsies or like a stabilization. But we feel like, a biopsy is really a tiny piece of tissue, and it's a really one section. So we really don't think that this is good enough to really see what's going on in tissues. So we have developed in my lab another way, and thank you for the Cystinosis Research Foundation who offered the skin confocal to my lab. So we are able to, we developed, this technology to be able to see a big piece of skin behind the ear, because this is where you don't have too much sun, exposure to sun, and it's easy access. So we scan the patients, and you can see here that after we do and they take like hundreds of pictures, like in a large thickness. So these give you a lot of data in once. And Alexis is here, so please, go and get scanned. We get collecting data to really try to one day hopefully validate this new technology to follow the patient with cystinosis. But so what we develop is that now we can quantify the cystine crystals. You can see the green points are crystals. And what I like this technology, because you can see that it's very sensitive, because this was baseline. You see all the baseline data, and for most of the patient, what you see is that when they do the transplant, the patient stops histamine, but the stem cell didn't have the time to go to the tissue yet. And what you can see is that all of them, you see an increase of cystine. So in few months, without histamine, you see that you have cystine crystals, building up. So, and we were able to catch that with this system. But what the good news is that after like a couple of months and 3 months, what we see is that a decrease of cystine, showing that the stem cells that carry the protein are now able to go to the tissues and do their job and decrease cystine. And so we saw that in most of our patients, except patient 4. I mean, when we saw an increase, and this is why he's coming back on cysteamine. But most of the patients are decreased, and the good news is that with the data we have so far, and the study we have so far, and that we published, we saw that the healthy subjects have the cystine crystals, including between 300 and 500, a maximum of 500. That's because we all have like backgrounds, it's like melanin. So they show like few background. And you can see here, for example, for patient 1 and 3, for whom we have the most data, so who have not been with cysteamine drug for many years, you see that they progressively decrease. So the stem cells are doing better and better. And you can see that patient 3 even have reached like a healthy subject level. And you can see that, I don't know if you can appreciate, I see really well the green points here, but I don't know if you see, this is a baseline. So the patient was under cysteamine, and you see that a lot of crystals are here. You see a decrease at 12 months, and the 36 months, so 3 years, are even much better. So I think that's, very reassuring, and we are really optimistic about that. We have also again, thank you for this, to the Cystinosis Research Foundation, who offered us also the cystine confocal to visualize and quantify the cystine crystals in the cornea. And so you can see here patient 1 with corneal crystals, at baseline. And you can see that for all the patients, we see a decrease of the cystine crystals, in the first few months. So there is like an impact of the stem cells, but after it's like it's very like stable. And the cystine crystals in the cornea are very special. Most of them are not into cells. So it seems like it can prevent the accumulation of new crystals, but the crystals that are not in the cells, they cannot remove them. So, it's stable, so that's a good news, but there is still crystals. And so that's why maybe a combination with some eye drop may be a good way to, really remove all the crystals. And for patient 1, his photophobia were very stable in 4 years. So what about the kidney function? So we have treated patients, who, as I said, 4 of them had kidney transplant, and 2 of them, patient 1 and patient 5, didn't have a kidney transplant. So here you have patient 1. So his kidney function after transplantation, where after a fluctuation after transplant, you see that it's quite stable for like about a year, and after it started to decrease. He had a kidney transplant at 2 years after a stem cell infusion, and now his kidney function is back to normal. For patient 5, we see that her kidney function decreased a lot, actually, at the time of a stem cell transplant. And we think that also she had also a COVID infection, and unfortunately, COVID has a really bad impact on the stem cells. So we think also that's why the vector copy number really decreased in this patient. And the nephrologist with who we work, she also thinks that it has a really bad impact on the kidney function. So you see that it was really a big decrease, and it continued decreasing. What we have to realize is that when we treat the adult patients, it's too late for the stem cells to stop the kidney progression into kidney failure. So we really hope that the next phase will be done in younger patients to see if we can really stop the kidney to deteriorate. The good news, though, is that the patient who had kidney transplantation didn't have, had a very stable kidney function, the same than before, or even better than before the stem cell infusion. So there is no detrimental effect of the stem cells on the kidney function if you had prior kidney transplantation. So that's the good news. What we have seen also is that actually because we do, so Dr. Troner, who has been years an expert in neurocognition function in cystinosis, is testing the patients, and we have seen actually in the patient improvement of the motor coordination and visual perception, and especially in the patient with who we have the longest follow-up, patient 1 and patient 3. And actually what we see is an improvement of muscle strength. So in cystinosis, a patient really developed muscle weakness, unfortunately, so you don't see an improvement of muscle strength. And in our patient 1 and patient 3, after like more than 3 years of stem cell infusion, with no cysteamine, we see a muscle improvement, muscle strength improvement. And I put this picture, because you really see the muscles now build up. So, it's really, I was like, can I show it? There is also a very weird, unexpected, endpoint that we observe in our patient, especially with patient 1. This is when we started to see that his skin and hair color change. It's normal that you have a change of hair color after a chemotherapy, but usually it's coming back after 6 months to a year. But after like 2 years, it was still like very darker than what it was. And actually, why this is important is because cystinosin is involved in the production of melanin. The melanin is the protein that color our hair and skin. And seeing this darkness, of the hair color and skin might be a good sign, showing that the protein is back. And that's why now we are decided, and I show, because, for the patient 1, we started later on because we didn't know. For patient 3, we started right in baseline, and you can see the kind of study we are doing now is like we take hair, and we have also a way to measure and biasly measure the hair color, and you can see, so lower is darker, and you can see that the hair color is changing. And actually, the dermatologists are very excited about that, and now I'm working with a team of dermatologists and also ophthalmologists, because we also see a change in the eye color, to really do a comprehensive study about this change in color. So in summary, we have fully enrolled the phase 1/2 clinical trial with 6 patients. We have a very good safety profile, which is great. We have seen a sustained hematopoietic reconstitution, and our vector copy number seems to be very stable over the years, so this is really good. But we have also seen a correlation of a vector copy number with cystine decrease and outcome. So with a vector copy number better over 1 is better. So patient 1 and 3, after over a year, they have really positive outcome. And patient 4 and 5 with a lower vector copy number, we have added 25% of cysteamine, and we believe that this, should be a good outcome too. So what I want to point out is that in the last 2 years, there were 6 hematopoietic stem cell gene therapy FDA-approved products only in 2 years. I mean, this is really the approval, as Betty said, the approval of gene therapy products are really speeding up. So in the last 2 years, there are 6 of them. And actually, 2 of them, so most of them are for blood disease, but 2 of them are for neurodegeneration, so brain disease. So it's possible, and we hope that, cystinosis will be part of this list one day. And so all this work was done as an academic institution with a funding from the CRF and CERM, and in 2017, AvroBio acquired the cystinosis program, but unfortunately, their company couldn't continue the program, and we are lucky that now Novartis is involved and got the program, so we have really good hope that they will be able to bring that to all the patients. That's the goal, and they are doing everything they can for that. I mean, that we have a table here, so please, don't hesitate to ask them all the questions you want. This is the time. And so I just, want to finish by, acknowledging everybody. I mean, you can see here that my husband and kids, have been part of the cystinosis family for many years, and they will be here tonight. So, this is part of our life. I'm really proud to be part of our community. And I want to thank you so much. Thank you, the CRF, for starting, this project. I mean, again, this would have never been able to start, especially with using hematopoietic stem cell for cystinosis. Nobody would have, trust me to fund this kind of project. But the CRF really believed in it, and as they said, they gave their biggest grant at the time. So thank you so much. We have been lucky now that we have leveraged this funding and got funding from NIH and CERM for the cystinosis research. It really takes a village to bring a gene therapy product from bench to bedside. So I want to thank, I know not everybody is there, but, I want to thank the people from my lab, the Cystinosis Stem Cell and Gene Therapy Consortium, Bruce Barshop, who is the PI, the clinical PI of a stem cell trial, all the medical personnel, of course, the family, the patients, the volunteers who really took the risk, without knowing if they would have any benefits of it, all the families for your support, all our collaborators, and of course, the funding. Thank you very much.
All right, thank you, Dr. Cherqui. I'm sure there are some questions in the audience, and you can ask them now or save some of them for the panel. But are there any questions at the moment? Yeah, let me get the mic.
Thanks for the talk. It's fascinating, exciting. Have you ever considered, or has it been thoughts about having maybe a second round of gene therapy eventually for patients that maybe aren't progressing as well with it?
So that's possible. You can do that. I mean, in theory, we could harvest the cells, and if we harvest the cells again, some of them would be already like gene modified and re-modify them. The issue is that it would be 2 rounds of chemotherapy or so. So after you have to, balance the consequences, the risk and the benefits. I think it's still a lot for the body, so I don't know if it's a good idea. But, I truly believe that the fact that even if the vector copy number is low, if we addition that with some kind of cysteamine, I think, it would be good because now the cells are, some of the cells still have a gene and they will still bring back to the tissue. So even if it's not perfect, it will be beneficial. So I don't know if we would take the risk of redoing it, but in theory, we could do that.
Any other questions? Yeah.
For the patients that didn't have as much success, are they taking eye drops?
So I have to ask patient 4. I don't think patient 4 is taking eye drops. Patient 5, yes, she's taking eye drops. Yep.
So what are the next steps moving forward? This is fantastic. What's the next move for testing, or do you have to wait on the drug company?
Yeah, so that's great. I mean, maybe, Daniel, do you want to comment? No? So the next step will be on Novartis. What we are doing is continuing to monitor the patients because this data will be important for the long-term effect. But now I know that Novartis is working very hard to get the next phase of a clinical trial started as soon as possible. They do everything they can to go. But, the FDA is very hard to work with. So, if it's not starting now, it's not because they don't want to. It's because the FDA wants more and more and more. So it's very difficult, So, but, I'm really confident that, it should start in the, hopefully soon. Yep.
Is the study just open to the KB deletion right now, or have you opened it up to different mutations?
Oh, if you see the first slide, they're all kind of mutations.
Okay.
So they are all different. And we have only one patient 6, who have a 57 KB deletion as of the homozygote state. So like 2 copies of a deletion. Otherwise, we have 1 copy of deletion, 1 over mutation. So there is no restriction.
Okay.
Thank you. Hi. Do the patients who have the therapy, stem cell therapy, and if they've been transplanted, do the kidneys have to be in a specific condition, or if they've had rejection, does that matter?
Yeah, no, they have to have a good kidney function. I think I'd have to remember the criteria, but it was more than 25% of kidney function or more than that. So they have to have a good kidney function to start with, even if they had kidney transplant. Yeah. All right.
All right. Thank you, Dr. Cherqui.